It's a pleasure to tell you about my group's work in this space of developing or determining membrane protein structures related to SARS-CoV-2 and also finding out about drug binding. And so my group employs solid-state NMR spectroscopy to understand membrane protein structure. And let me just see. Okay, so and one class of membrane proteins that we have been particularly interested in is viral porins. The prototype member of this family of proteins is the influenza M2 proton channel. So viral porins in general mediate virus budding and assembly. They also conduct ions. And it's this ion conducting property which is targeted by antiviral drugs. And these proteins tend to be pretty small, less than 100 amino acid residues. They tend to form oligomeric helical bundles in the virus envelope or in the cell membrane in which these viruses assemble. And because these are small helical proteins, they're pretty difficult to crystallize because there's just not enough soluble domain in these proteins. And being so small, they're also difficult to detect by cryo-electron microscopy. So solid-state NMR is a natural spectroscopic technique to give us high-resolution structural information about these proteins. So influenza M2 is a canonical member of the viral porin family of proteins, but there are other ones, and SARS-CoV-2 is the system that I want to tell you about today. So in the SARS-CoV-2 virus, we know that there are three structural membrane proteins: the spike protein, the membrane protein, M, and the envelope protein. This E protein is the smallest and is the most mysterious because it's too small to be seen by the traditional structural biology techniques. So what do these three membrane proteins do? Well, they orchestrate their actions in different time points of the life cycle of the SARS-CoV-2 virus. We know that the spike protein brings the virus into the cell by binding to cell surface ACE2 receptors. And once inside, the viral RNA is replicated, transcribed, and viral proteins synthesized. And the new viral proteins and RNAs are assembled in this compartment of the cell called ERGIC, or ER-Golgi intermediate compartment. And here, the E protein functions to make sure that the new virus buds into the ERGIC lumen, after which the new virus can be then exocytosed out of the cell. And so the first function of the E is to help to bud the virus properly. But the second function, let me just try to get this off my screen. The second function is that it conducts ions, and it conducts cations, non-specifically sodium, potassium, but the calcium conducting activity has been associated with the triggering or stimulation of the inflammasome of the cell. And so this is of particular interest to us. Now, hexamethylene amiloride, the small molecule shown here, has been known to be a weak inhibitor of the E protein, but exactly where it binds, how it binds, and so on was not known. And of course, there's interest in developing more potent inhibitor of the E protein. So my lab, as I said, develops and applies solid-state NMR spectroscopy methods to study membrane proteins as well as other disordered systems. I don't know what happened here, but anyways, so in NMR, what we do is to detect the oscillatory response of atomic nuclei that carry non-zero spins to resonant irradiation of radio frequency pulses in a large magnetic field. Now, the frequency response of the atomic nuclei contain information about the chemical structure of your molecules. And that's why NMR is the eye of chemists. You figure out the basic chemical structure. But in structural biology, what we're interested in is as much about chemical structure as the three-dimensional structure. And that's because the peak intensities encode information about the internuclear or interatomic distances through the dipolar coupling, which is distance dependent. Now, in solid state, when we study membrane proteins as bound to liposomes, multilamellar liposomes, lipid bilayers, we have to get high-resolution spectral information by doing something called magic angle spinning. So we put our sample, hydrated liposomes, in a cylindrical rotor, which is tilted by 54.7 degrees from the magnetic field, and we spin around this rotor axis. Under that condition, we can then get liquid-like high-resolution spectra that contains information about the chemical structure as well as the three-dimensional structure. So this is the basis, this magic angle spinning solid-state NMR is the basis of all our spectroscopy. So now let me tell you how we apply solid-state NMR to determine the structure of SARS-CoV-2 E protein. So early on in the pandemic, we quickly expressed and purified the E protein's transmembrane domain, which is about 30 residues, by using a sumo fusion tag. And we can express this to multi-milligram quantities in M9 minimal media with carbon-13 and nitrogen-15 enrichment. And then we can cleave the sumo tag and purify everything by HPLC. And so we get reasonable yield to do our spectroscopy. And with solid-state NMR, what's really nice is that we can incorporate the protein of interest into lipid membranes that actually mimic the composition of the biologically relevant membrane. And the ergic membrane contains the regular phosphatidylcholine, PC, PE, and negatively charged PS lipids, cholesterol. In addition, it also includes phosphatidylinositol. So we basically made a mixture which is representative of the ergic membrane composition. And our first experiment was done under neutral pH conditions. And as you can see from the very first set of 2D spectrum, here are 2D nitrogen-carbon correlation spectra, we can already resolve very well the residues in the transmembrane domain. You can practically just in 2D see about 2/3 of the signals from the TM domain being resolved. And so by assigning these chemical shifts using more extended three-dimensional correlation experiments, we can sort out the chemical shifts of the protein, carbon and nitrogen chemical shifts. And what these chemical shifts tell you is that the transmembrane domain is largely alpha helical based on these chemical shifts, except at the very end near that arginine, then there's a few residues that are more disordered. And so this defines the basic backbone conformation of the protein. And the fact that the line width is so narrow also tells you that the E's transmembrane domain is very well ordered in phospholipid bilayers. Now, backbone conformation and the monomer conformation is not enough to determine the full quaternary structure of this kind of helical bundles. And to really sort out the entire structure, you need to know interhelical distances, and you also would like to know the helix orientation in the lipid bilayer. And with solid-state NMR, we can determine both. And I want to first show you our strategy for measuring interhelical distances. Now, you can imagine these helix separations are roughly going to be on the order of about 10 angstrom or a nanometer. And traditional carbon-13 or M15-based NMR techniques, it's hard to measure distances to that range because the magnetic dipole moment for carbon and nitrogen is not very strong. And so this is one reason, the key reason that my group became interested in recent years to develop fluorine solid-state NMR methods to measure nanometer distances. Fluorine is the second highest gyromagnetic ratio spin 1/2 nucleus among all stable spin 1/2 nucleus. It is only slightly weaker in the gyromagnetic ratio, the gamma, from proton by about 6%. So based on this property, we can predict that when you measure distances between fluorine and some other nuclei, you can go much longer than the traditional C13 or M15 NMR. So my group and these dipolar couplings depends on distances in this inverse cube fashion, but also depends on the gamma. So without going into details, I'll just say that in the last 5 or 6 years, my group has developed a whole toolbox of 2-dimensional and 3-dimensional fluorine-involved distance NMR methods that can allow us to measure to about 1 to 2 nanometer range. And these involve both carbon fluorine as well as proton fluorine distances. And the reason that there are 2-dimensional and 3-dimensional experiments is because we would like to measure many distances at the same time relatively quickly. And so let me just illustrate our workflow using the SARS-CoV-2 problem as a practical example. From the literature, before our work and after the first SARS-1 epidemic in 2003, there was some biochemical data that suggest that E is maybe a pentameric system. And so what we did was to mix C13 and fluorine labeled transmembrane peptide in a one-to-one fashion and measuring the interhelical carbon fluorine distances. And we do so in a two-dimensional experiment, correlating carbon-carbon chemical shifts so we can resolve the peaks. And then we turn on fluorine pulses to cause dipolar dephasing to the nearby carbon spins. And by subtracting two spectra, we can then obtain the delta S difference spectrum that indicates all the carbon spins in the neighboring helix, which are adjacent to the fluorinated residues. And where do we fluorinate? We tag a fluorine on the aromatic phenylalanines that are native to the transmembrane peptide. So in this fashion, we can then measure which carbon sites are close to the phenylalanine residues, and these quantitative distances then became important constraints for the confining or constraining the helical bundle structure. So this approach, this line of experiments are called REDOR. So we, in addition to these REDOR experiments, also can figure out or measure the topology or the packing of the helices by using water transferred and lipid transferred experiments. And here the idea is to transfer, for example, magnetization from the pore water molecules to the protein protons and then detect that through the protein carbon-13 signals or M15 signals. And so in that way, we can sort out which residues are pore-facing versus lipid-facing. So here is an example of a 2D water edited or water transferred to the nitrogen carbon correlation spectrum, where shown in blue are well hydrated, high intensity from the water magnetization, and in red are the dry residues that then are lipid-facing. We can also switch the initial magnetization source to transfer from lipids, and that in that way we can figure out that, for example, these phenylalanine residues are more lipid-facing because they get their magnetization from lipid protons more readily. So when we combine the backbone conformation from chemical shifts, the quantitative carbon fluorine interhelical distances, as well as these water-facing and lipid-facing topological constraints, we can then obtain a high-resolution structure of the E transmembrane peptide. And this structure was solved at low pH in an ergic mimetic membrane. And if you look at this tight, relatively vertical, not very tilted helical bundle, you will conclude that this is probably corresponding to a closed state of the channel. The pore diameter ranges from about 10 to 12 angstrom. So we don't think that this state can conduct ions. And you can look at details of the structure, finding out, for example, that the pore-facing residues are the hydrophobic, methyl-rich, leucine, valine, isoleucine, etc. And there are 3 regularly spaced phenylalanine residues in the middle of this transmembrane segment, and their side chain is interesting. They seem to form this pi stacking conformation across the helix interface. We think that this configuration or structure will stabilize the helical bundle. And we have further experiments that show that under different conditions, for example, going to low pH, adding calcium, that these side chain conformations will change that potentially loosen this middle portion of the helical bundle that may support then ion conduction. So this is the first structure. And when we quickly solved the structure in 2020, using the literature information that this helical bundle is pentameric, we got questions from the community. Are we really sure about the pentameric structure? And here are some of the literature papers that showed gel data and analytical ultracentrifugation. And as you can see, these gel bands are not that clear. They can sort of count to sort of 5, oligomer number of 5, but it's not certain. And so that's the reason we get all these questions. And in addition, you can also ask whether the lipid bilayer bound oligomer state is the same as what's found in detergents. And so to sort out these questions, we decided to directly measure the oligomer state by NMR in these ergic mimetic membranes. And our approach is, again, fluorine NMR. And here the strategy is a so-called Codex experiment. And in a nutshell, what Codex measures is the spin diffusion effect from one fluorine to another if there are multiple fluorines in relatively close proximity within about a 2 nanometer range. And if there is such an oligomer formation, that there's fluorine magnetization transfer, then we can run this CODEX experiment to detect a spin echo. And if you have a trimer, the equilibrium intensity of the echo should be 1/3. And if you have a pentamer, it should be 1/5. Basically, what you're measuring is the initial, the fraction of the magnetization that remains in the initial site after this full equilibration of the magnetization transfer. So that's the concept of the experiment. So using then this CODEX experiment, we incorporated a single fluorine into the E transmembrane peptide and one of the native phenylalanines just at the para position. And as you can see here in this echo intensity measurement, when we have waited long enough to see the magnetization equilibration, we found at about 2 seconds or 3 seconds, the value echo intensity to approach 0.2, suggesting pentamers. But if you go longer, wait longer, you find the echo intensity drop below 0.2, suggesting higher oligomer species, which is interesting. And we decided then to dilute the membrane, the protein concentration by a factor of 2. And at this point, we found that the equilibrium echo intensity plateaued at 0.2. So if you have higher concentration, then we can see more than the basic unit of pentamer, you basically have additional spins that maybe 2 pentamers approaching each other. So the fact that this more than 1 pentamer can approach each other in this ergic mimetic membrane is interesting. We decided to sort out which lipid might be responsible for that. And we can show that if you remove cholesterol and phosphatidyl inositol, then even at the high protein concentration, the equilibrium value is around 0.2, while having the PI and cholesterol is the reason for this further drop. And so basically what we learned is that the SARS-E oligomerization, the basic unit of oligomer formation is pentameric, but if you have PI and cholesterol in your membrane, and then you can have clustering going on, and that would then allow you to see more than 5 spins. So this is a point of interest that we're investigating further. Now the question is, well, okay, if you have a closed state at neutral pH, what does it take to form a conducting open or activated channel? And so Joao in my lab decided to study the E protein, the transmembrane construct at low pH in the presence of some calcium. And the reason for having these 2 ions is because ergic is acidic compared to the cytoplasm, and it is calcium rich. Its calcium concentration is about 2 or 3 orders of magnitude higher than in the cytoplasm. And we can show using the water transferred protein signals that if you add protons, make the sample acidic, and add some calcium, or have one of these 2 ions or have both, then the water magnetization transfer to the protein is much faster, indicating that you basically have a more water accessible pore, and that's basically a physical definition of an open state. So we then decided to measure or determine the structure under the condition of acidic pH in the presence of about 20 millimolar calcium. And here's just a 2D carbon correlation spectrum between the neutral pH condition and the low pH calcium bound condition. And shown in blue and green is basically higher water transfer. And that indicates, again, you have a more water accessible pore. And so this is how we think about it. At low pH, in the presence of calcium, you have a wider pore. And so then we conduct the same kind of distance measurements, interhelical carbon fluorine distance measurements. Now we also add proton fluorine. Again, the protons will come from the adjacent chain, and all of these Redor experiments give us very quantitative measure of distances. And we supplement those with more traditional carbon-nitrogen-based NMR measurements, where here the idea is we mix C13 and M15 labeled peptides, usually in a 1-to-1 fashion, and we measure 2D correlation spectra and measure the cross-peak intensities to figure out which carbons and nitrogen from adjacent chains are close together in space. Now, in addition to interhelical constraints, I want to tell you a little bit about helix orientation measurement. And here, this is obviously a very independent type of structural parameters, really orthogonal. And the idea here is that we put our ETM pentamers into a slightly different lipid bilayer, POPC, POPG, which is low viscosity. And in that bilayer, we check to see if the helical bundle is undergoing uniaxial rotational diffusion. And if it is, then we can measure the motionally averaged NH dipolar coupling for every backbone amide group. And that backbone NH dipolar coupling under this rotational diffusion condition basically tells us about the helix orientation. So we check to make sure that indeed in this low viscosity and very fluid membrane, this whole helical bundle is moving as seen by these data. And then we extract NH dipolar couplings and then convert them to the helix orientation. All right, so what we find is that in the open state, most of the peptide is slightly tilted, only about 5 degrees from the bilayer normal, except for the C-terminal, about 1/3, it's slightly more tilted. And so this is a very useful constraint to give us, allow us to calculate the atomic resolution structure. So now shown in pink is the pentameric E at the low pH, calcium present condition. And you can see how this 5-helix bundle looks quite different from the neutral pH tight helical bundle that I showed you earlier. The N-terminal 2/3 really looks like a helical barrel of some sort with more space for water molecules that is needed for ion conduction, while the C-terminal 1/3 of the transmembrane domain is slightly more tilted, actually kind of pinches closer. And this we later figured out in separate measurements that these actually are not so alpha helical, they're more 310 helices. And what does the structure tell us about the potential ion conduction mechanism? It turns out that in the N-terminal segment, about 7 or 8 residues, there is a number of polar residues. For example, this glutamic acid, and then there's several threonines, and then there's asparagine here. And based on the low pH structure, we find that this glutamic acid can adopt 2 types of configurations. In one, it reaches out to the next helix threonine to form a potentially hydrogen bonding that may be useful for chlorinating calcium. And then in a separate, a second set of conformations, this glutamic acid points its carboxyl group to the N-terminal or the surface water, and this could be useful for recruiting protons to then relate into the channel. So in the C-terminal end of the transmembrane domain, we find an arginine residue, which is actually goes into the channel pore, which is a bit surprising, but we then can rationalize this to then, this inward-facing arginine could then make the channel pore so polar that it can reduce the energy barrier for pulling or conducting ions across the hydrophobic middle of the entire channel. And this arginine side chain conformation is quite different from the closed state where, based on our calculation, it's pointing to the lipids forming potential salt bridges with the lipid phosphates. Now, determining the structure really is for 2 purposes. One is to understand the fundamental mechanism of ion conduction, and then the other is really to try to facilitate drug binding, drug development, and then understanding the drug binding mode. And so this HMA is a molecule that is known for a while, and we are fortunate to have medicinal chemists from both UC San Francisco, from Bill Delgado's lab, and from Rutgers University Jun Wang, that provide us with these very nice isotopically enriched drugs, whether it's C13 and 15 labeled at the guanidinium moiety or fluorinated at the hexamethylene ring end. Both are very useful for NMR-based determination of the drug binding site. And so what we do is to have orthogonal labeling, different types of labeling motifs between protein and drug. And so I just want to give you a brief idea, how do we sort out the binding site? And these drugs, this kind of HMA drug molecule, as you can guess, has affinity for both the protein and lipids because it's pretty amphipathic. And so the first question is just how many drug molecules actually target the pentamer. So the binding stoichiometry is one question, and the binding site is the second question. And so if you imagine having a sample that is 1-to-1 protein to drug or 5-to-5, then if the binding stoichiometry is 1 drug to each pentamer, then we can determine that by observing the drug signals and as dephased by the protein. So if you have an excess of drug, then you'll find only 1 out of 5, let's say, of the drug will be affected by the protein. And once that stoichiometry is sorted out, then you can turn around to protein detection. And with that, you can find out whether that one equivalent of drug is bound centrally inside the pentamer, or perhaps is it bound to the surface, because this will make a difference to the observed protein dephasing. So this is the concept. And so here I want to show you data with C13, combining C13 and 15 labeled drug with a fluorinated protein. And we observed the drug signal, C13 and 15, as dephased by the protein. And we have 2 samples. In one, we have 2 equivalent of drug added to 5 protein monomers. And when you detect the drug, you can see that the read-or signal goes down to about half, approaching half, which means that only 1 out of 2 drugs actually is in contact with the protein. And when we have another sample, which is 5-5 drug to protein, you can see that the drug signal decays only to about 80%, again saying that only 1 out of 5 drugs is in contact with the protein. So this allows us to conclude that the stoichiometry is 1 drug per protein in the lipid bilayer. So now we're able to switch to protein detection. And here we're running experiments detecting the protein amide nitrogen signals and amide proton, and we have a 5-5 sample. Then we detect, in this case, the drug dephasing. So the drug is fluorinated, and the protein, we're looking at the nitrogen signal. And as you can see, the protein signal only dephased to about a very low, only 0.9 or 0.8, which means the majority of the drug actually, or most of the protein, is not close to a drug. So this drug cannot be centrally located. So more detailed analysis then allow us to dock, but these are the crucial features that allow us to say that there's only 1 HMA bound per pentamer, and moreover, that HMA is not inside the pore experienced or causing dephasing to all 5 monomers. Rather, it is sort of peripherally bound at the protein lipid interface or helix-helix interface. And these are the Haddock result, docking result, as constrained by all the measured distances. So the conclusion is that this HMA, the amiloride compound, inhibits E by binding to the protein lipid interface and helix-helix interface, tightening the channel. So this information is very interesting because this HMA binding mode, inhibition mode, is quite different from amantadine binding and inhibition of the influenza M2 protein, which we learned about 15 years ago using sort of similar strategy, but not fluorination. We used a deuterated drug at the time, and it's very clear that amantadine actually goes inside the channel, M2 channel, inhibiting by directly occluding the proton conduction pathway. So SARS-E and possibly other coronaviruses, E proteins, may have this very different inhibition or mechanism against their amiloride compounds, or for the amiloride compounds. So with that, I would like to conclude. So I acknowledge various people along the way, but Shiva and Matt started the SARS-E project in 2020, and then the work is followed up by Joao, Noah, Aurelio, and Eva, and Alex Sherbakov really contributed in developing the line of fluorine distance NMR techniques. And we are fortunate to have the collaboration with the UC San Francisco group, many people from the medicinal chemistry side that I didn't list here, that provided some of the HMA drug, as well as the newer AViDD drugs that I didn't show you, and Bill DeGuado's lab for long-time collaboration, and June Wang. And thank you for your attention.